Well, students, we have now moved into Chapter 5, in which we're going to build upon the knowledge that we have gleaned from Chapters 1, 2, 3, and 4. And primarily in this chapter, we're going to focus on information that we learned and hopefully can apply from chapter number 4. Keep in mind that chapter 4 refers to carbon compounds, the unique characteristic of carbon being that it can form 4 individual bonds. We can do this as far as carbon is concerned, 4 single bonds, we can have 2 double bonds, we can have triple bonds, different varieties. And because of the diversity of carbon, we're able to see a multitude of compounds that are formed. And the chapter that we are dealing with here in Chapter 5 is a concept known as macromolecules. And macromolecules are going to play a huge role, especially in the organism. And in this chapter, in this particular lecture, we want to cover 3 main ideas. I want to talk to you about polymers, I want to talk to you about carbohydrates, and I want to talk to you about lipids. So that's going to be the 3 things we cover in this lecture, and then in another lecture we will look at the last 2 components of this particular chapter, which focuses on proteins and nucleic acids. So let's go ahead and get started by dealing with the opening portion of this chapter, and that is talking about polymers. Now when we talk about polymers, I don't want you to get too burdened with the word. It may be a new word to you. But this word just simply means many parts. And so when I talk about a polymer, we're going to be talking about a molecule that is large, but this large molecule is made up of smaller parts, and we call those smaller parts monomers. And another thing to keep in mind is, just like with the alphabet, we have a lot of words. Just pick up any dictionary, and you'll see that there are a lot of words formed by only 26 letters in our alphabet. And we're going to see very much the same thing in this discussion, where we're actually going to see a multitude of polymers that are able to be formed by a small set of monomers. As you go through your notes, you're going to see that this actually is taken from about 40 to 50 monomers that are very common, and then there are some rare ones in there as well. Now as we go through this lecture, we are of course going to be focusing on what are called macromolecules. And these macromolecules come in 4 basic varieties. We have what are called carbohydrates, lipids, proteins, and nucleic acids. And of these 4, there are 3 that actually come from what we call monomers that develop or build into these polymers. And those will be the carbohydrates, the proteins, and the nucleic acids. So our lipids are a class that aren't actually formed by monomers. But we will get to those in just a little bit. So as we move through this, again, I'm going to highlight some of these terms. We may move through a few of these slides very quickly, but just keep in mind to jot down any questions that you might have and bring those to class, and we will begin discussing those on Monday. So polymers, again, these are classes of macromolecules that are formed by chain-like molecules called polymers. Polymers are the large guys, and then we have repeated units of smaller molecules that we call monomers. And these monomers are actually going to have functions of their own, and when you add them together, you're going to get new functions that are going to occur on the polymer region. Now before we can get into a discussion of the individual macromolecules that we have, we need to understand a little bit of how these things are going to actually form. And so keep in mind that we have this concept, this idea of monomers. Now monomers are small molecules that are actually going to build into larger molecules. And so as we look at this, if we're going to take some small monomers, or these short polymers, in essence, where we would actually be talking about smaller chained polymers, and we want to make larger polymers out of them, we're going to go through a process that is called a condensation reaction or a dehydration reaction. In fact, we can also call this dehydration synthesis, because you're actually building things, you're adding things together, and anytime you build things, you're going to be dealing with some type of combination or synthesis reaction. So as you go through this, notice what we've got here is we have a monomer providing a hydroxyl group. And if you've studied your functional groups, the OH, remember, is the hydroxyl group. It is going to add together with a hydrogen from another monomer, and when we take an OH and we add an H to it, we get H2O, so we actually will form water, or we produce water. And in order for this to happen, there's going to have to be some energy supplied. And we'll talk about this in the next couple of chapters, but this energy that is being required is because we're going from small molecules into large molecules. And in order for us to do that, we're going to have some energy that allows this to happen. The key thing to remember is that we're going to be forming water in order to take small polymers or even individual monomers and make a longer chain polymer from it. On the other side of that, we have the reverse of this particular reaction that is called hydrolysis. Now, in hydrolysis, what will happen is you take the polymer or the large compound and you can actually break it down. And the way this is done is we use water. So water is produced in a condensation reaction. Water is going to be used or broken apart in a hydrolysis. Hydro meaning water, lysis meaning to split. So in essence, we're going to split water into its component parts, hydrogen and hydroxyl group, or hydrogen and hydroxide, and they are going to be added to the corresponding portions of a polymer, and now you're going to end up having smaller chain polymers, or if we want to go down on an individual level, we can actually break this down into the monomer level. So just make sure you keep that in mind. Again, when we deal with these reactions, you've got condensation reactions or dehydration synthesis. We're going to form water, pull water out of the molecules, and in essence add things together. This is going to require energy. In hydrolysis, we're going to use water to split the molecules apart, and when that happens, we're going to actually release. Dehydration synthesis would be a combination or synthesis reaction. Hydrolysis would be a decomposition reaction because we're breaking something large into smaller component parts, which is going to release energy into the environment. And as I've mentioned before, an immense variety of polymers are formed from a relatively small set of monomers. Somewhere between 40 and 50 common monomers and a few rare ones will actually allow us to build into these large macromolecules. We want to start with carbohydrates. Carbohydrates are going to actually include three different classes. We have what are called monosaccharides, what are called disaccharides, and what are called polysaccharides. So we start off with the simple one. Simple carbohydrates are called monosaccharides. The word mono means simple or single, saccharide meaning sugar. So we've got one sugar or simple sugars. Disaccharides are double sugars. The word di meaning two, the word saccharide again meaning sugar, so double sugar or two sugars. And this is going to happen through a condensation reaction. We're going to take two monomers or two monosaccharides, and we're going to put them together through dehydration synthesis or this condensation reaction, and we're going to build a disaccharide. And then the third class is going to be the polysaccharides, which are the polymers, and these polymers are going to exist because we take individual monomers or individual monosaccharides, add them together, and we're going to actually be able to build from that these really long-chained carbohydrates that we call polysaccharides. Monosaccharides are the simplest of the class, and you notice here that generally, now this isn't always going to be the case, but in general, there's going to be a formula that is CH2O. And what this tells us is that carbon's going to be present, hydrogen's going to be present, and oxygen's going to be present. So from the name, carbohydrate. And it's a 1 to 2 to 1 ratio. So we're going to have 1 carbon to 2 hydrogens to 1 oxygen ratio. So here's an example. Glucose. Glucose formula is C6H12O6. You have a 1 to 2 to 1 ratio as pertaining to the chemical makeup or the chemical formula of this particular monosaccharide. We are also going to be talking about carbonyls. We're going to be talking about hydroxyls. And if you remember those functional groups from the last chapter, bear in mind that when it comes to a carbonyl, you've got two different kinds. You have what are called ketones and what are called aldehydes. And this is going to play a key role in the naming and also the functioning of our monosaccharides. If you have a sugar, a monosaccharide, that has the carbonyl group somewhere in the chain but not at the end, then it's going to be called a ketose. The word, the OSE ending is for sugar, so a ketose. If it ends in that carbonyl, then we're going to have an aldose or an aldehyde sugar. Monosaccharides are going to be classified by the number of carbons they have. So if we're dealing with glucose, which happens to be a 6-carbon sugar, we call it a hexose because it has 6 carbons in it. 5 carbons are called pentoses. 3 carbons are called trioses. And so just make sure you keep in mind all of those prefixes that hopefully you've learned along the way. We also have enantiomers, if you remember that from the last chapter. These are going to be isomers, if you will, of these monosaccharides, these sugar compounds that are actually going to have the same basic functional groups, but they are oriented in such a way that you can't superimpose them, and because they're not able to be superimposed, we have different molecules there. They have different spatial arrangements. So glucose and galactose would be examples of that. They're both 6-carbon aldoses, so they're going to be monomers that actually end in the carbonyl, but their spatial arrangement is going to allow them to have different functions and actually be different molecules. So here we have a graphic representation of that. I won't spend too much time on this. Trioses, pentoses, and hexoses. And then you also have your aldoses, which are in the yellowish orange, and then your bluish color are your ketoses. And again, you can look at those and see that if you're an aldose, you're going to have the sugar, the carbonyl actually at the end of the sugar, and if it is a ketose, you're going to find the carbonyl in the middle of that chain. The most important one for us that we're going to be dealing with throughout this year is glucose. Glucose is a monosaccharide that is used to fuel cell power or to fuel cellular work. When we get next couple of chapters, we'll talk about cellular respiration, and the big sugar that we want to talk about there, the big monosaccharide, is of course glucose. But these sugars can also be used to synthesize other materials as well, not just disaccharides and polysaccharides, but they can also be used for the production of amino acids and fatty acids, which means we start moving into the protein and the lipid category. We have also what are called disaccharides. Disaccharides are 2 sugars that are put together through dehydration synthesis. So again, we're pulling water out, taking 2 small things and adding them together. And this is done by what is called a glycosidic linkage. And all a glycosidic linkage is the process in which we are going to remove a hydroxyl, we're going to remove a hydrogen, we're going to put them together and form water, and when that water forms, we're going to have a larger molecule. Every time we add a monomer to a monomer, every time we add a monosaccharide to another monosaccharide, we're going to have a glycosidic linkage, and that glycosidic linkage is going to form these longer chained polymers, disaccharides, and ultimately then into polysaccharides. And 2 important ones that we will be dealing with throughout this is called maltose. Maltose is going to be a disaccharide that is formed when you take 2 glucose molecules and put them together. One that is much more familiar to us is this one. This is sucrose. Sucrose is actually formed by taking glucose and adding fructose to it, and sucrose is table sugar. So the sugar that you put on your cereal or anything you put table sugar on, you're actually using a disaccharide or sucrose. Thirdly, we have the polysaccharides. Polysaccharides are polymers of hundreds and even thousands of monosaccharides joined together by glycosidic linkages. So we're going to have these dehydration synthesis reactions, these condensation reactions occurring in which water continues to be pulled out. And keep in mind, every time we do this, one new water molecule is formed. So every time you form a glycosidic linkage, you're going to have a water molecule that is formed. Polysaccharides are used for energy storage inside of the organism, and we have several different varieties, and we want to talk about those. Plants have some, animals have some. They do have some similarities, but of course, since they're in different organisms, they're also going to be different. They're going to have different functions in the individual organisms. And some are going to serve as building materials for the cell, and in some cases, it'll actually serve as building material for the entire organism. So here's the first one for us to talk about, one that is probably fairly common, which is starch. This is a polysaccharide that is composed entirely of glucose monomers. And so what we mean by that is you're taking glucose and glucose and glucose and glucose and just continuing to add them together. And as you add them together, you end up with these starches. Two varieties of this are what we call amylose and what we call amylopectin. And you can see in the diagram and also in your notes that these have different structures, therefore they have different functions. This one, amylopectin, is a little more complex than the amylose, but they are both types of starch. And again, these are all glucose molecules. When we get to animals, we have another type of polysaccharide that is very similar. It is only glucose, and this is glycogen. Glycogen is inside of animals. It's very similar to amylopectin when it comes to its branching. It's not a straight chain like you find in amylose. It's very highly branched, and this is the type of polysaccharide that humans as well as other vertebrates are going to store inside of the liver so that in case we need to have some glucose released into the environment, we're able to break that glycogen down and release the glucose into the environment. And then we also have, when it comes to our polysaccharides, they're going to be built from a variety of monosaccharides, but glucose is of primary importance. There are so many things that this one particular monosaccharide can form just by the individual linkages that it has. So if we look at our slides here, continuing on, we're going to see that there are some differences here in the way these polysaccharides develop because of two different chains that actually occur inside of glucose. Glucose can either form what is called an alpha ring or what is called a beta ring. Now don't get too lost on this, but simply all that's taking place in an alpha ring versus a beta ring is how the glucoses line themselves up. So for example, here we have starch, which is a linkage of alpha glucose monomers, and what you're going to notice here is the hydroxyl groups are on the same side. So as you look at these, they are basically the same thing that is linked together as they go across. Now compare that to cellulose, which is another important polysaccharide in our plants, especially when it comes to their cell walls. Notice that as we go across, you're actually going to see that we have the hydroxyls on the bottom, and on the next molecule it's on the top. So as you go across, for every glucose that is added, the one that precedes it and the one that succeeds it is actually upside down relative to the one that we are discussing. When it comes to our polymers, they're built with alpha glucose to form helical structures. Those that are beta are going to form straight structures, okay? And this is going to offer a wide variety of bonding characteristics because of the way they are formed. Continuing, there's also what is called chitin. Chitin is a polysaccharide that is used by our arthropods. These would be insects, spiders, and crustaceans. And its job is to basically provide a strong support structure on the outside of these soft-bodied organisms so that they have an extra layer of protection. So make sure you jot down any questions with regards to our sugars, to our carbohydrates, monos, dis, and polysaccharides. I want to conclude the lecture, this first lecture, with a discussion on lipids. Lipids by nature are hydrophobic. The word hydro meaning water, the word phobic meaning fearing. And so we're talking about a class of macromolecules that are fearful of water. What that means is they have no affinity for water. They don't mix with water very well. Now there is one that is very unique, and we'll talk about it in just a moment. We have what are called fats, phospholipids, and steroids. So we want to look at those over the next few moments. Lipids are an exception. This is the one class that do not form they do not have polymers. So we're not monomers that are actually built into polymers like we have with the others. They have no affinity for water or very little, and this will come into play when we deal with what we call phospholipids. And this is because since they are very heavily dominated with carbons and hydrogens, they and these covalent bonds, they're non-polars. And since they're non-polar, they're not going to mix well with water because like dissolves like. When it comes to dissolving, like dissolves like. So polar dissolves polar, non-polar dissolves non-polar. They're very diverse, and they have a lot of different forms and functions. So we're going to start out first by looking at what is called a fat. Fats are going to be formed by two different molecules that come together, glycerol and fatty acids. So here's a diagram of that. Here is our glycerol skeleton, and that glycerol skeleton is going to attach to a fatty acid through dehydration synthesis. So we see the water being formed. You've got the hydrogen here, and you've got the hydroxyl group here. They come together, they form water, and now we form this linkage that occurs between the fatty acid chain and the glycerol component. If we want to talk about a triglycerol, or what is also known as a triglyceride, this is a class of lipids that we really regard as our fats. So we have this chain, if you will, of or skeleton of glycerol attached to these three fatty acid chains, and that gives us the triglycerol molecule. These different fatty acids come in two varieties. We have what are called saturated and also what are called unsaturated fatty acids. And in dealing with these compounds, there's going to be some unique features, some unique functions regarding this. So we have here, you're looking at some butter, you're looking at margarine, solids at room temperature, and this occurs because we have single bonds between the carbons. If you're looking on your slides here, you will notice that you have these single bonds in between the carbons, which means they are saturated or completely surrounded by hydrogens. Because of that, because they are held so closely together, we end up forming a bond that leaves the product as a solid at room temperature. If we put some double bonds in there, we're going to have some bending that takes place. And if you remember from chapter 4, when there's a double bond, the orientation is pretty much limited into the amount of movement that it can have, so it's not able to be pulled as tightly together. And because of that, your unsaturated fatty acids are going to remain liquids at room temperature, and you get this kink that is actually formed because of those double bonds. And everywhere there is a double bond, that is one less hydrogen that can actually be on that individual carbon atom. So if you have two carbons that are double bonded, that's two hydrogens that are now unable to occupy those spaces. So we call those unsaturated. If we're dealing with our diet, we would much rather our diet be heavier in unsaturated than we would in saturated fats. Our saturated fats, again being solid at room temperature, they can contribute to cardiovascular disease, especially this atherosclerosis or hardening of the arteries because of plaque buildup. Try to imagine what it is on the outside being what it is on the inside. So if it's a solid on the outside, it's going to solidify on the inside, which means you're going to have limited space inside of your arteries. The heart's going to have to pump even harder to course that blood through your system. Unsaturated fats are going to be liquids, and so you're not going to have the issues inside of your arteries and your blood vessels as you would with the solids or the saturated fats. Major function of fats is energy storage. In fact, they will store more energy than you will find in a carbohydrate. A gram of fat will store twice as much energy as a gram of a polysaccharide. So they are real good energy storers. We're dealing with a type of macromolecule that is an energy reserve. It also provides cushioning for vital organs, and it provides insulation for organisms as well. So we don't want to be too heavy on the fats that we put into our diet and put into our body. However, they are important for us to have. A second class are called phospholipids. Phospholipids are a very unique type of lipid because as with triglycerides, they are completely hydrophobic. They want nothing to do with water at all. Try as you may, you will not be able to mix a fat with water. A phospholipid, on the other hand, has what we call a hydrophilic portion as well as a hydrophobic. So part of it is water-loving, part of it is water-fleeing or water-fearing. Makes it very unique, and it also allows it to be a major component of our cell membranes. If you remember from life sciences earlier, we're dealing with what is called a phospholipid bilayer. And the structure of a phospholipid is very similar to a fatty acid, with one major difference. You still have a glycerol. Okay, so the glycerol is still present here just as you have inside of the triglyceride. However, here's where the difference comes in. You've got two fatty acid chains, but you also have what is called a hydrophilic phosphate group. And the phosphate group is actually going to be polar because phosphate by nature is a negative ion, and so it's going to be attracted to the polarity of the water. So it will actually face the water molecules, and the hydrophobic tails will face one another because they are trying to escape mixing from the water. And so if we deal with this, this is a picture of the phospholipid bilayer of our cellular makeup. So you have water on the inside, water on the outside. Remember, we're 70 to 95% water inside of our cells. And so if this, let's say that this is inside the cell, this is outside the cell, and the blue represents the water. The phosphate heads, which are hydrophilic, will point towards the water inside and as well as outside the cell. And then the hydrophobic tails are going to point to one another. They are going to point away from the water, and this is going to allow things that are soluble in water to be dissolved inside of the water. Things that are not soluble in water but are soluble in this organic material in the phospholipid bilayer will actually be able to dissolve here and then move into our cell. The third thing, as we conclude this lecture, are our steroids. Now, our steroids are a very unique class of lipids as well. This is their main structure. So the one we're looking at here is cholesterol, but remember in class that I built for you testosterone and estradiol, which are sex hormones, but these sex hormones in the male and female respectively are steroidal. So their backbone is going to be this steroid, and you can see that you have these 4 carbon rings that are connected. And then there's a variety of functional groups that can be added to this to give the different types of steroids that we find. Cholesterol is a very important one. It's a support structure, provides support to the cell. Cholesterol being found inside of our cell membrane keeps the cell from collapsing upon itself. But we don't want to have too much of it. It is important. It helps to produce or to manufacture the sex hormones in males and in females, but we want to make sure that we limit cholesterol in our diet, because as our cholesterol level goes up, it can contribute to cardiovascular disease in much the same way as the fats that we talked about earlier. So to recap, what we have looked at in this lecture has been monomers coming together through dehydration synthesis to form polymers. Polymers can break down through a process of hydrolysis and form the individual monomers. Then we have our carbohydrates, our first class of macromolecules. Carbohydrates are going to they come in three varieties: monosaccharides, simple sugars; disaccharides, double sugars; polysaccharides, many sugars. And every time we put two monosaccharides together, we have a polycytic linkage, and those linkages, or glycosidic linkage rather, these glycosidic linkages lead to the more complex types of molecules, disaccharides and polysaccharides. And we talked about our lipids, three classes of lipids. We got the triglycerides, saturated and unsaturated, dealing with single bond. If they're all single bonds in our carbons, we got saturated. If we got double bonds in there, then we got unsaturated fats. We've got our phospholipids, and we've also got our steroids. Be sure to write down any questions that you may have. Bring those to class on Monday, and we'll try to cover anything that you were unable to understand.